I'm speaking with Gary Nabel, the Chief Scientific Officer of Sanofi. Thanks for joining me today, Gary.
My pleasure, Vince.
These chats are part of our textbook of virology, and the one I'm having with you is for the chapter on vaccines. And I'd like to start off by having you tell us a little bit about where you were born and raised and educated.
Well, my family history is pretty straightforward, although there's a little history before I was born that probably was formative in my life too. But I was born in Connecticut, Hartford, and grew up in the Hartford area, went to high school there, went off to college to Boston and did my undergraduate work at Harvard and my MD and PhD at that institution as well.
Who did you do your PhD with there?
With Harvey Cantor, who's an immunologist who was one of the discoverers of T-cell subsets, which he did shortly before I joined his lab. And my family ended up in Hartford after they moved up from your fair city, from New York, and they had been immigrants from Europe, from Poland. So both of my parents were actually Holocaust survivors who were liberated by the Americans at the end of the war and came over and were welcomed into this country. And so we've been grateful for all the opportunities we've had.
So what got you interested in science to begin with? It sounds, the way you describe it, that you had a linear progression, but was there any thought about it very early on in your life?
Yeah, you know, actually I didn't plan to go into science. I was originally quite interested in government. I liked figuring out how you could do the most good for a lot of people, and in the naivete of my youth, I thought government was the way to do that. But I had an exposure to science when I was in high school that was pretty formative. I was given the opportunity to join an NSF program that was being run by a college professor from Amherst at a local private school. It was called the Program in Biochemistry. And we spent 6 weeks studying biochemistry and doing lab experiments. And it was something really about that program that catalyzed the interest in me. Not just me, I would say easily 80% of the kids that I met there, because they were from around the country, went on to do either an MD or a PhD, and most of them ended up doing some research. And I think it was in that environment of being around peers and being excited about the problems and being able to talk about it and do things with your hands. And see results that gave you, feedback in a relatively short time. That was pretty much what started me on that road. It wasn't an automatic switch the day I finished with that program, but through college I kind of switched back and forth between government, science. And it was actually eventually the head tutor in the government department who convinced me I should do science, because he said, and medicine, he said, if you major in government, you'll never be able to do science, but if you major in science, you'll always be able to come back and do government. So.
Good advice, good advice. Why did you decide to get a MD and a PhD?
For me, it was the natural thing to do. I love people, and I am a pretty social and I'd like to think caring person. So medicine seemed like a good fit. But I really also love the science and the ability to translate from what you learn in science and bring that to people. To me, was always sort of my calling. And even though at times I've done some pretty esoteric science and drilled into some pretty basic questions, at the end of the day, I still find it probably the greatest reward when I can take that knowledge and bring it back and apply it to people.
So let's complete your training. You got your MD, PhD at Harvard. What did you do after that?
I then went on to do my residency at Brigham and Women's Hospital, and there I did a special track for, they called it a hemidoc program. It was for people, mostly MD PhDs, who wanted to keep doing research but get trained in medicine, and they allowed you to split, that's where the hemi came from, your time between the laboratory and the clinic. In reality, it's actually maybe 2/3, 1/3 you do more clinic, because you have to spend the time in the clinic to get trained. But it allows you to keep things going in the lab and then to have a quick start back when you return. I continued in that setting to work in Harvey Kanter's lab at the Dana-Farber Cancer Center, but it was actually during that period that I kind of arranged my postdoctoral experience, and that I did at the Whitehead Institute with David Baltimore. And so I went straight from my residency to David's lab, spent about 3 years there before I moved to take my first faculty position at the University of Michigan.
Which is where I saw you at one point in our careers. What did you work on in David's lab?
Well, the most notable thing that I did in David's lab was it was the very early days of HIV, and we were looking at transcriptional regulation of HIV, and that was the work where we discovered actually that the transcriptional activator, the switch to activate HIV transcription, is NF-kappa B. And so we demonstrated that NF-kappa B was newly discovered at the time. It was in 1987 or so. And we were just beginning to learn what the motifs looked like. And I noticed when I looked at the enhancer region of HIV, there were a couple of kappa B sites. So we did the experiments to show binding, and then we did the mutations to show that if you knocked out those binding sites that you were unable to activate expression of HIV. So that was probably the most notable piece of work out at David's lab. But I'd say more generally, I was interested there in molecular mechanisms of viral activation and the immune response to viral infection. So that's really, I think, for me, where the connection to virology and vaccines really started, was when I was with David.
So that was going to be my next question. You spent a good part of your career at the Vaccine Research Institute?
Vaccine Research Center.
The Vaccine Research Center of the NIH, working on vaccines of various sorts. And how did you get interested in vaccines? It sounds like you started in David's lab, but is that something that developed as you were faculty?
Yeah, it developed. I was always interested in HIV and AIDS research, obviously because of the, the devastating effects of the infection, which we literally, grew up in.
Right.
I still remember seeing some of the very first patients presenting in the hospital, and so wanted to, and the biology of what we were seeing at the time was so unlike anything we'd ever seen before. It just drove that curiosity. In the beginning, most of my work in the HIV area really focused on the basic side, looking at transcriptional activation. We cloned subunits of NF-kappa B. But then what I started to do is to see if we could use our knowledge of viral gene expression and the viral gene products themselves to try to block replication. So we actually performed some of the very first gene therapy studies that were directed at suppressing HIV replication. This was when I was at Michigan. We actually developed vectors to transduce T cells where we could make dominant negative forms of the viral genes, in particular Rev. We could show actually in lymphocytes from patients that suppressed viral replication, and you could get selective outgrowth of those cells. So I'd been working in that area, was very active in HIV research, and meanwhile the epidemic was storming along, and it became pretty clear that it was vital to get an AIDS vaccine. It was going to be the only way to really, provide a long-term solution to the problem. And there was, in 1998, a speech that Bill Clinton gave about developing an AIDS vaccine, and in the speech he said, I want to propose, or I'm going to use a presidential directive to build a vaccine research center dedicated to the development of an AIDS vaccine. Originally, I was just a advocate and assistant trying to help them find a director for the center, and recommended all sorts of my good virology friends who, for various reasons, decided not to do it. And one day I get a call from Harold Varmus, who was the NIH director at the time, and he said, would you consider being the director of the center? And I said, after I stopped laughing, I said to Harold, I said to Harold, well, really, I'm pretty well set with what I'm doing. I loved Ann Arbor. I saw no reason to leave. But he said, well, come down and visit. And so I came down, visited with Harold, and then with Tony Fauci and with Rick Klausner. The three of them had organized, actually, the inception of the center. And the more I talked with them, first of all, they were all very persuasive in terms of both the scientific challenge and the human need for a vaccine. And then I started to think, well, the work that I had been doing on the molecular genetics and gene transfer was every bit as complicated as developing a vaccine. And I began to realize that if we were able to make progress on the on the vaccine problem, the impacts would be so much greater. So for the same amount of time spent, the net impact would be, a lot more widespread. So that's what really got me thinking about it. It was also a tremendous opportunity in that we had a brand new center, brand new building. The NIH leadership was really giving me incredible support. I think certainly Harold, while he was there, Harold left probably another 2 years later. But then really over the long haul, it was Tony, who I would say was an unbelievable supporter of both scientifically, personally, in terms of resources, advice, everything. And so it made my job both fun and easy. It's good that the job was fun and easy because the scientific problem is quite daunting. And so we put together the center, got all of the pieces into place. We have a phenomenal group of investigators, and we were really able, I think, in the course of the almost 14 years that I was there, to make some significant progress on the vaccine. Haven't solved the problem, still lots to do. But I do think we helped to move the field, because there are obviously many people involved in AIDS vaccine development, but we helped to move it to a new place. And I think we now have a variety of options open to us that we never imagined we would when we started the work. And we've learned a lot of very basic biology about the virus through that effort as well. So that was really a wonderful experience, and it was awfully hard to leave the NIH.
So can you look back at your years at the Vaccine Research Center and pick a key experiment or a key result that really stands above all the others?
Well, the ones that are there's one or two that stand up, at least in my mind. I would say one of the most memorable moments or memorable discoveries that we made was when we used our knowledge of the structure. I would say, Peter Kwong was really the person who was on the front lines and solved a number of structures that enabled us to understand the mechanisms of entry of the virus, understand the mechanisms of immune evasion, and to even understand the molecular and structural basis of virus neutralization. And we worked very closely together and used that knowledge to really probe the immune response, particularly the B cell response. And I'd say one of the highlights was when we were able to use those structures, genetically modify the highly conserved sites, go in, and then with John Mascola, who was doing a lot of the antibody neutralization work, the three of us were able to go in and identify some of these exceptionally broadly neutralizing antibodies. The first one was called VRC01. And at the time, it was really quite remarkable because it was really, we went 10 years in the field where we said and thought we had broadly neutralizing antibodies. There were 4 of them, and there were no more in actually more than 15 years. And now when we look back at those 4, we would call those not broadly neutralizing at all. They were pretty poorly neutralizing. These new ones are hitting 90+% of viral strains, and they recognize the virus through the receptor binding site, through the CD4 binding site. So when those data emerged, that was really pretty striking, and because we never imagined that a single antibody could have that breadth. And more than that, that antibody actually came from a person. So it was a proof of concept that those kinds of antibodies can be generated, and they can be generated in people. And if we can find those in a natural immune response, it certainly gives a lot of impetus for developing a vaccine. And I would say maybe equally important, as we've gone along, we've now realized that those antibodies themselves are tools that we can use for prevention and treatment and possibly even cure. So we're still involved. Some of this now at Sanofi and through my lab there, and some in collaboration with the VRC still, to try to see if we can shift our thinking to use them in more of an industrial mode. Can we expand them up, make them at the scale you would make them, formulate them in a way that they would have long half-life? So it's presented some new opportunities for treatment. So that has to clearly be one of the highlights. I'd say the others have had to do with applying the same approach to the development of vaccines for other infectious diseases. That understanding of the molecular biology, the understanding of structure, the understanding of the immune response to those viral components. And the other examples that I've been very gratified to see move forward, one is an emerging problem, which I'm hoping our efforts will provide a solution to, and that's chikungunya virus. So we were able to make a very nice VLP of chikungunya virus by just expressing the structural proteins. And again, we brought that through animal models, non-human primates. We've recently written up the first phase 1 human study where it elicits very, very nice neutralizing antibody responses to chikungunya. So being able to take the model and apply it, and we're doing the same thing for universal flu vaccines, where we can take the same approach, design immunogens, and there we can elicit some of these broadly neutralizing anti-stem antibodies. So I'd say that HIV was the leading edge, and then it then opened up opportunities that we could more readily develop and apply to some other viral vaccines.
So the original charge of the center was an HIV vaccine, but you were able to move into others as well. Was that your idea? Was that part of the original inception?
When we started up the center, we held a series of meetings with scientists in the field, and we developed a mission statement, a series of goals, and we always said that the primary emphasis would be HIV, but the overall goal was the rational development and design of vaccines. And so it was always an intent to sort of use that knowledge and apply it to wherever it made sense. And yeah, I think we stuck with that. I think the wisdom of that approach wasn't my idea, was really a collective conclusion from those retreats, was that there would be other microbial threats, and that just in, you know, what we used to say at the VRC in terms of the response to HIV is that if we got the vaccine one day sooner, it would mean 7,000 less people infected. That's how many people are infected each day with the AIDS virus. So there was an urgency to what we were doing. And when, in the days following 9/11, when we had the anthrax attacks, and then when we saw, you know, the H5, and then when we saw SARS, and then when we saw, you know, more recently MERS, you know, we've seen this play out time and time again. So the idea was to develop systematic approaches based on what we were doing to with HIV that could then be applied in other cases.
So the idea of having a vaccine research center, I think, is a great idea, but of course other people in academia and industry also want to develop vaccines. So do you have a collaborative view with them, or do you work on your own?
Oh, it's very collaborative. You know, it's interesting. We did a lot of things at the VRC to allow us to span the spectrum of what I would call early vaccine development. That means fundamental virology, fundamental immunology, translational virology, immunology, going up the species scale and into humans. That required us building a pilot plant, which we literally did from the ground up. It required us doing a GLP lab where we could measure human immune responses. But even with all of that, you're only at the starting line for actually implementing a vaccine. So all along the way, you know, there's a value chain in research, and so at the very basic level, our scientists were collaborating with academic scientists in structural biology. You know, the Scripps group, Ian Wilson, Dennis Burton, that group, Michelle Nussenzweig in New York for the B-cell biology. Then as you got into the clinical part, we would do phase 1 studies, but we would really very much depend on the NIH networks, clinical trials networks, the HVTN. So Scott Hammer from your institution led our efficacy trial. And then you clearly have to constantly be building bridges to industry.
Sure.
Because if your manufacturing methods aren't consistent with what they can do, then you'll have to start way back at the beginning.
Right.
So yeah, it was very interdisciplinary, and really it was a lot of fun because you learn so much from the collective wisdom of those groups.
So you've been in the field a long time. Can you give us a sense of how technology has changed in a way that, say, benefited vaccine development?
Oh, I think it's been critical. I think that the things that we can do now based on the technology that's available just we wouldn't have even dreamed of when we started. So, you know, where can I start? You know, certainly genetics and genetic sequencing goes without saying. I think that came online relatively early in HIV research because of the diversity of the virus. But there was a lot that had to be, you know, modernized to really get the throughput and the rigor, and to avoid a lot of the learnings and artifacts early in the days. I think clearly, for example, next generation sequencing, particularly with regard to interrogating the immune response, because what we now can do when we identify a new antibody and we can sequence it, we can look at all of the relatives that that evolve within one individual, between individuals. We can do the phylogenetic analysis of those molecular clones. Right. And that teaches us, that provides a roadmap in terms of vaccine design about where we need to hit the immune system to trigger those responses. The structural biology, you know, in the early days, you know how long it took to get a structure. And now, you know, I remember recently in Peter's lab going from an idea for a sequence to the structure in a matter of weeks. So it's been transformative. And another major advance, I think, was what Michelle Nussenzweig pioneered, which was the single-cell isolation of immunoglobulins from single cells by PCR. It, you know, it doesn't sound like much because it's just PCR, but, you know, compared to what we used to do with hybridomas, or with, obviously, with, you know, even some of the more recent EBV transformations, you know, we can plow through, you know, a repertoire in a very short period of time. The B-cell biology, understanding the differentiation of B-cells so that we can pick out the subsets that are going to be activated, that's critical. So I'd say between structural biology, genetics, our fundamental understanding of immunology, and I would say too, even on the production side, on the technology side, our ability to get into place producer lines and the quality control and the methodology for scaling up production, all of them have really propelled us into a new era.
So I think you're unusual in that you've worked in academia, you worked for the government, and now you're working for a private corporation. What's different about all that?
You know, I felt very fortunate to have each of the experiences that I did, and they're all different in different ways. I think in academia, you know, I felt like I was essentially in a little cocoon where I was being protected and nurtured and allowed to work on the science that I really enjoyed working on. Times were a little different then in that the grants were a little easier to come by, and it gave you the freedom really to pursue the things that you think were most important to you. And I think that ability to work that way, and then also to have that opportunity in academia to connect, you know, with the biotech world, then can amplify and make your life interesting in terms of being able to translate at least on a smaller scale the research you're doing into a more practical space. The NIH is very different in the sense that you still are given, I think you're probably given more access to at least a sustainable support. So you're given a longer runway and a little bit more freedom to take longer term risks. And that's something I think that we've really lost nowadays in supporting academic research. But and I also think that with the diversity of what NIH can support, you're able to make connections and to develop some of the kinds of infrastructure, like the pilot plants I was telling you about, and analysis labs that can really facilitate translation. Having said that, there still are limitations to the NIH, and I would say after coming to industry, that's been instructive as well. I think you know, the science you apply to the problems no different, your judgment is no different, but what is quite different, I'd say there are probably two fundamental differences in industry that stand out. One is that unlike the NIH or in academia, where your research support comes to you and it's a fairly known quantity, maybe a little less in academia now, but in industry you actually have to earn your research support. It's not given to you, and so if you don't make something that's of value to somebody, you're not going to be able to continue your research. I would say the other thing, and it goes probably hand in hand with that, is that the real power in industry is the power to scale things, to manufacture at a level where you really can get out there and have an impact on the world. And that's a tremendous opportunity. It's also a tremendous responsibility, because doing it rigorously and making the choices about the things you will move along versus the things that you're going to have to take a pass on is something that you've got to be very smart about. And what strikes me, and I say this both from my NIH experience, but also from the industry side, because we still work with the government. We work with the Gates Foundation, very much involved with the Gates in terms of the polio eradication programs, is that there are certain things that industry would not and could not do on its own, and there are certain things that government would not and could not do on their own, and philanthropy as well. But when you put them together, these public-private partnerships, there really is a remarkable synergy. And so I think that opportunity, particularly when applied to things like vaccines or other preventive measures or other transformative kinds of cross-sweeping technologies, then you can really have a major impact. So I view the different roles that I've had over the years as being each of them very special. I think it's given me a great appreciation of the ecosystem of innovation, the academic, the government, private, biotech, philanthropy. And so I can see where each can excel on its own, and I can see where there are really wonderful opportunities to work together.
Gary, of all the things you've done in your career, can you tell us one that you think has had the most impact on science?
Well, I think it gets back to what we talked about before. I think it's developing the strategies for understanding the interaction between the virus and the host, and then using that to rationally design. My hope is that I don't think we've crossed a finish line. I think we're probably off the starting line and maybe somewhere early in the race. But my hope is that either the antibodies themselves or the knowledge we've created in developing, specific vaccine candidates will reach fruition, will actually develop into products that can be given to people and that will help them. And then, it'll be interesting to watch, because I'm not sure, for example, whether HIV will have its greatest impact first, universal flu, or chikungunya. And somewhere along the line, I probably left out Ebola, which is rearing its ugly head again. I would be delighted if any one of those, crossed. I think we've crossed major hurdles and really advanced to significant milestones, but it's a marathon, not a sprint, and we're not there yet.
I think I know the answer to this next question, but I'm going to ask you anyway. If you hadn't been a scientist, what would you have done?
That's a good question. I probably would have gone into government. Although I will say, in retrospect, I did very much enjoy medicine when I was doing it, and I could have been pretty fulfilled just being a country doc.
Somehow I'm not sure about that. I'll take your word for it. So readers of this book may be interested in a career in science, and as we've discussed today, science is exhilarating, but it's also getting difficult to do. What kind of advice can you give them?
Well, you're right. I think the science is changing in terms of how we do it and the tools we have available. I think the most important thing, and I think particularly for most of the readers of this book, since I assume they're relatively early on in their career, is I still think the old principle of just follow your passion, particularly while you're training. You know, I think that study questions that are, you know, of fundamental importance that you really want to know the answer to because you think they're essential for getting to that next level. And I think if they learn the discipline of doing science, identifying a good problem, developing a strong hypothesis, understanding the ways to test that hypothesis, and then to, you know, learning how to write papers and communicate the science to others. If they master those basics, the rest will unfold. I think one of the issues that are facing young scientists right now, and I think one of the challenges of encouraging people to get into the field, is that they tend to look at people who are very senior, and they tend to think, I'll never be able to do that, so why bother? But they forget that nobody ever got to where they were in life by jumping over the Grand Canyon, right? It was by marching on these trails and looking at these, you know, little streams that they could hop over, which eventually got them to the point where they built these programs. So don't try to do it in a day. Follow your passions, become well-grounded, and, you know, and pursue the opportunities. You know, keep your eyes open for the opportunity, but don't force it when it's not there. So I still think it's a privilege to be able to do it. I still think there's, you know, no greater satisfaction, at least for me, than to discover something that no one has ever understood before. And then I think you can amplify on that by then taking it to people, helping people. So I think it's a great career. And I also think in terms of the trajectory of research where we are, I think we should also take a step back and say, look, times are tight now, but times have been tight before. And there is waxing and waning in the research cycle. We're in a drought period now in the research cycle, but that's not going to last forever. And not to say we're not going to have to be creative and thoughtful and intelligent about how, you know, the more senior folks solve those problems, but better days will come, and we should make sure that our you know, lifeblood for science doesn't get turned away because of these what I think will turn out to be short-term problems.
I've been speaking with Gary Nabel, the Chief Scientific Officer of Sanofi. Thanks so much, Gary, for talking with me today.
It's my pleasure. Thank you.